very much indeed to the organisers and yourselves for inviting me to come and talk about lung infections and cystic fibrosis. I come from Southampton, and I look after adult cystic fibrosis there, and we have about 250 patients, but I'm pleased to say I've visited Athens and the adult cystic fibrosis department there. And so I'm going to talk about some new ideas that we have about lung infections in cystic fibrosis. As you know, having it and having relatives with it, the most troublesome thing is the fact that you get chest infections, and those chest infections are treated by antibiotics. And that could be thought to be reasonably straightforward, but it isn't, as I think I'll talk to you about today, or mention to you about it today. And there's some new words that are coming along, and one of them is the microbiome. Instead of microbiology, we're talking about a community of bacteria, not just the one Pseudomonas that was mentioned today, but a whole family of bacteria. So let's just start. This is the sort of information as doctors and patients we get, and you can see that here are the down below are the bacteria that we get on culture: Staphylococcus, Pseudomonas, and oral flora. They're the sorts of organisms you get in your mouth. And on the right-hand side there are the antibiotics that we choose to treat those infections. And that's the information we get and you get, or have done so. Number one says that we might be able to treat this with flucloxacillin, and number two, we suggest maybe ceftazidime and another aminoglycoside, tobramycin. So probably on this, we're quite happy with ourselves, and we have this information, and we say to you, well, I think we'll give you some ceftazidime and some tobramycin, and that should do the trick. And what we do know is it does do the trick. That if you give an antibiotics for an infection, then on the whole, it goes away. But then we start getting other sorts of, on other occasions with other people maybe, that they get more than one Pseudomonas. They're growing yeasts, which is a fungus, and they're also growing these upper airway bacteria. But can you see the R there? The R means it's resistant to those antibiotics there on the side. And so we're wondering what to do about that. And what we tend to do is probably give you the same thing. A bit of ceftazidime and tobramycin, see how that goes. But that's not so satisfactory, in that the information we're getting in the laboratory isn't the sort of information, it doesn't fit with what we're doing. So there's a mismatch. And then look along at this one. So you've got two species of Pseudomonas there on the left, and some oral flora again. But look at all the R's down there. So what do we do in that situation? Do we give people ceftazidime and tobramycin as well? Or sometimes we do. And so there is what do we know? We know that antibiotics work for respiratory infections and cystic fibrosis. But we know that the information that we're given by culture isn't really accurately reflecting what we're doing. So we then thought, right, well, is it to do with the fact that when we culture the sputum, we just pick out one or two colonies? What if we took all of the sputum, as we're doing here, and added all the little discs for antibiotics? Would it be we got better information if we didn't select out the colonies, but actually just put the whole sputum on the plate and two sets of antibiotics to see if we could better identify which antibiotics are going to work for you. Now, why is that important? That's important because we want to get it right. Having antibiotics for a long stretch of time is really difficult, and we want to make sure we give you the antibiotics in a way that doesn't give you side effects. But when we did this and put all the sputum on the plates and put the antibiotics next to them, 2 or 3, we couldn't work out, it didn't make any better difference than doing it as we had done traditionally. That was in Southampton. And this is a very famous paper here by somebody called Juliet Fauraker in Cambridge. Now what they did, we took the approach of trying to do, to put everything on the plate and seeing whether that would work. She took the opposite approach. And what she did was in great detail look at all the colonies of Pseudomonas on a cultural plate and pick them out and see what those individual colonies, if they responded to antibiotics. And what did she find? She found that in one patient there was probably 4 different types of Pseudomonas, and those 4 different Pseudomonas had different profiles of antibiotics. So it was enormously complicated. So, okay, what's the man doing in the tulip field? Well, if you have one infection with a bacteria, it's very easy to see, and if you have a urinary tract infection or meningitis, we can pick out that infection and give you the right antibiotic. But cystic fibrosis isn't a tulip field. Cystic fibrosis is a beautiful herbaceous border, lots of different flowers and weeds all over the place. It's a very different situation. It's a mixed situation. So why am I showing you a glacier? That's because with all these doubts about how more to give you specific treatment, we started to talk to environmental biologists, people who worked in the soil, because in the soil you have lots of mixed bacteria, and it's not like the tulip field. And as a glacier recedes and melts, the soil underneath at first is sterile, no bacteria there at all. But as it melts, bacteria come in one by one in a very ordered way. And that's just like what happens in cystic fibrosis. So as a child, usually, the first bacteria you find are Staphylococcus. And then after that, Haemophilus. You get a little bit of fungus in there, and then Pseudomonas, and then probably a more complicated Pseudomonas. And that's what happens in the soil as the glacier recedes. So we thought these people probably know a little bit more about the situation we're in, not the tulip field, but the beautiful herbaceous border, and we'll use their techniques. So this is what we normally do. We take the patient, and there in that green pot is your sputum. And we put that sputum on a culture plate. Well, what grows? Well, it grows the pathogens or the bacteria that that culture plate is meant to grow. But do you know that 90% of the bacteria we can't culture? That plate won't grow 90% of the bacteria. So not understandably, in a cystic fibrosis situation, there is very little correlation between the sensitivities of the bacteria and what we grow. So what we do is we take the patient, give them the sputum and grow a particular bacteria that we think is there, and we give antibiotics for that particular bacteria, but we're missing huge amounts of information about all the other bacteria. So maybe that's why there is this mismatch between the antibiotics we give you and how you respond. So what I'm coming on to is these techniques that the soil biologists taught us. And they, looking at their situation, which is the soil, which is incredibly complex, what they do is something very different. They knew that in the genes of all bacteria there is a special region called the 16S that every single bacteria has, and that's really good luck for us. Because if we take the sputum now and extract the DNA from the sputum and look at the bacteria, look at all the bacteria and their DNA, we will be able to find we found a way of looking at all the bacteria and not just the ones that we think are there. So there is no cultural stage, and it avoids what we think is there is what actually is there. And we sequence the DNA and you can do it with the RNA, but actually we do the DNA so that we can look both at what's there, and the RNA will tell us how active those bacteria are. And what we call this is TFRLP analysis. And I'm just going to show you, because I don't want to make it too complicated, but if we add a little signal onto the 16S bit of the bacteria, and chop it all up and amplify it, at the bottom there you can see different lengths, and the different lengths are related to a specific bacteria. So we did that. And this is what you get. You get a gel. So you put all the bacteria at the top and then they precipitate down, and the longer ones go down further and the shorter ones go at the top, and there's a little library that you can say that length means that bacteria is present. But this is no good if this technique doesn't also detect what we can find by culture. So we compared what we cultured and with what the new technique showed. So you've got culture on your left, and on the right you've got doing it in a new way. And what did we find? Well, we found some really interesting stuff actually, that culture sometimes over-represents bacteria. So if you culture something, you get more Staph aureus, but maybe there wasn't very much Staph aureus there in the first place. Also, there are species that we can't culture because they're anaerobe, that is, they're in the dark. They're very difficult to culture, and we don't set them up for culture, like Prevotella and Stenotrophomonas multifilia. So we get much more information if we use this, and it's not biased. And in fact, sometimes with culture, you get an exaggerated result with some bacteria like Staph aureus there. So this is the same information I gave you on the previous slide, shown in a different way here. In that there, obviously, you get more bacteria if you use the molecular technique, and if you're just relying on the cultural technique, you get less. That's obvious because there's no example. Pseudomonas is probably not a successful strategy, and that we shouldn't go for the big words like eradication and removal, because we're not probably going to be able to do that. And that gives you side effects. Affects your hearing, affects your kidney function. So we've got to think of other strategies of giving you the right antibiotics for what is a mixed situation and not the man in front of the tulip field. We may need to do lower doses for and less lengthy courses. And guess what? The best way presently of assessing whether a course of antibiotics is working is to ask you, okay? To ask the patient, do you think the antibiotics I'm giving you at the moment are working? How long do you need them for? That's the best we've got actually at the moment. So what we need to know, I think it's, I've given you lots of questions today, or a new way of looking at things is, what is the significance of these newly described bacteria? And actually, what is an infective exacerbation? And what are the antibiotics we're giving you doing? And we need to think more fluidly of the choice of antibiotics, the dose and the length, bearing in mind these antibiotics gives you side effects, and thankfully we're all living longer, so that we need to preserve the antibiotics and the amounts of antibiotic doses we can give you, particularly the aminoglycosides. And so basically, what this is a community of bacteria, and not the man sitting in front of the tulip field. What the situation in cystic fibrosis, and it's not just cystic fibrosis, it's bronchiectasis, chronic obstructive airways disease is a mixed situation, and we have to think of strategies to affect the mixed situation, which isn't a sort of eradication or total removal, but of influencing what these communities of bacteria, all these plants that are all interdependent on each other, how can we influence it positively? Okay, so that's what I was, well, that's something this slide is.
Thank you very much for this presentation. Once more, it showed that CF is a complex disease, and we're getting always learning more and more about the disease, but also learning more and more how many questions still have to be answered. But we're happy to have people like you and the other experts who help us to find a reply to all these questions. So any questions or remarks for Dr. Caroll?